Hello students, this is chapter 1. This is a very introductory chapter to get us started here in our microbiology course. I generally like to choose interesting or sometimes funny cover slides to get us started, usually something that's relevant to the chapter. In this case, this image that I chose was from an article that came out a few years ago, and it was a microbiologist, and she had an 8-year-old son, and she wanted to see what was lurking on his hands when he came in from playing outdoors. And so she had him press his hand onto this Petri plate that contains agar, which is a growth media that will allow bacteria to grow. And so after incubation, this is what resulted, and it was really incredible, and I kind of think it's beautiful. You might think it's disgusting, but it's really neat to see the variety of bacteria that grew from this child's hand. So each little circular growth, or big circular growth in some cases, that you're seeing here is a different type of bacteria. So each color, each size, each shape, each texture, we can determine is a different bacterium. So you see there's some yellow colonies. So, each of these yellows, they're likely the same bacteria. The oranges are likely the same. You have a variety of whites, different whites, off-whites, beige, etc. So again, if they look different in size, shape, color, texture, then generally that indicates it's a different bacteria. So I don't remember if in the article she quantified these colonies, but it would be possible. It would take a little bit of work, but you could actually quantify and kind of determine how much bacteria was on the child's hand, how many different kinds of bacteria were on the child's hand. But even without doing any quantification, you get the idea. You can see that there's a lot of bacteria out there, and we can pick them up on our hands, and that's why hand washing, of course, is so important. But let's get on with this chapter now. So in this chapter, or in this semester, in this course, I should say, we will be covering microorganisms. So what are microorganisms? Well, if you break the name apart, they are organisms that are microscopic or too small to be seen without a microscope. And so we will mostly focus on bacteria in this class, but we will also briefly cover these other types of microorganisms as well. Now, I don't have a picture over here to represent all of the different types, but here's bacteria in picture A. I don't have a picture for archaea, but we'll see one on an upcoming slide. Here's fungi in picture B. Here's a protozoa in picture C. Algae in picture D. Viruses in picture E. The red here is HIV, actually. And then I don't have a picture for the last one, but again, we'll see it on an upcoming slide with the multicellular animal parasites. So those are the types of microbes that we will be covering in this class, but definitely we will focus more heavily on bacteria throughout the course. So what are bacteria? Well, bacteria are prokaryotes or prokaryotic cells. You've probably learned about prokaryotes and eukaryotes before, hopefully. Prokaryote really means pre-kernel, which the kernel is referring to a nucleus. So pre-nucleus. They do not have a nucleus. One way, and if you check out the Amoeba Sisters, I do have links to lots of good YouTube videos and animations on our Canvas page. If you check out the Amoeba Sisters, they give a nice little rhyme for remembering prokaryotes versus eukaryotes. They say pro, no. So prokaryotes don't have a nucleus. Pro, no nucleus. You do. Eukaryotes do have a nucleus. So bacteria, they are single-celled prokaryotes, and that means they don't have a nucleus, they don't have membrane-bound organelles, but they have something that is very unique to them called peptidoglycan. And this is a substance only found in bacteria. No other living organisms possess it. It makes up their cell walls. It's a very important substance that we will cover more in the future because a lot of antibiotics, including penicillin and penicillin's relatives, specifically target peptidoglycan to kill bacteria. Bacteria divide by binary fission. This is a little bit different than mitosis. It's actually simpler, so we'll cover that in more detail in a future chapter. And we will also cover in a future chapter bacterial metabolism because it is a little bit complicated. They actually have the most diverse metabolic capabilities. Bacteria can derive nutrition from organic chemicals. That's like what we do, proteins, lipids, carbohydrates. But what's unique about some bacteria is they can use inorganic chemicals to derive nutrition. Things like, think of the periodic table, sulfur and nitrogen and things like that. Yes, we need those in our diet, but we can't get nutrition. We don't get calories directly just from eating those elements. And often we can't even utilize them in that elemental form like bacteria can. Some bacteria can also do photosynthesis. We won't really be focusing on that in this class because in general, photosynthetic bacteria are not pathogenic. They don't cause disease, and our focus will be on disease-causing microorganisms. Some bacteria are able to be motile with flagella. Moving on to archaea. So archaea are also prokaryotic cells, but they don't have peptidoglycan like bacteria do. Some of them don't even have a cell wall at all. And what's interesting about archaea is they are found living in extreme environments. These include the methanogens, which produce methane gas, extreme halophiles, which live in extremely salty environments, and extreme thermophiles, which live in extremely hot environments. And again, we only touch on archaea very briefly in here, kind of usually as a compare-contrast to bacteria and to eukarya, because archaea generally are not known to cause disease to humans. And again, our focus in this class will really be on disease. So fungi. Fungi are eukaryotes, so they do have a nucleus. They do have membrane-bound organelles. Some eukaryotes have cell walls, some don't. In this case, fungi do have cell walls, usually made of chitin or other types of polysaccharides. Fungi, as far as their nutrition goes, they only use organic chemicals for energy, and they absorb them across their fungal bodies or across their cells. There is multicellular forms of fungi, like you might be familiar with, molds and mushrooms. And then there's unicellular forms of fungi, which are the yeast. We will cover fungi in more detail in chapter 12, but just a little preview of some of the structure of fungi. You can see that molds consist of masses called mycelia, and those mycelia are composed of microscopic filaments called hyphae. And those hyphae lead to spore-forming structures, some of which are called sporangia. There's some other terms as well that are used for these spore-forming structures. And it's these spores that are the way that fungi reproduce. So the only thing we have labeled on this diagram is sporangia, but these other threads down below that lead to the sporangia, those are actually the hyphae. And if we just talked about this whole mass of fungi or mold, you would call it a mycelium. So protozoa are also eukaryotes, and they only use organic chemicals for their metabolism as well, and they're either absorbing them across their cells or they may ingest them with a mouth-like structure. They can be motile with either pseudopods, which is what we're seeing in this picture here. This is an amoeba that's extending out its pseudopods. Or they can have cilia, or they can have flagella, or they might be non-motile with none of those structures. A lot of protozoa are free living, but there are some that are parasitic. And I should mention that also about bacteria and fungi, and just mention that while we will focus on disease-causing organisms mostly in this class, it is important to know that the majority of microorganisms are not disease-causing. Majority of them are out there, free living, whether we're talking about bacteria, fungi, protozoa. Majority of them, they're out in the environment, they're free living, they're not causing any problems. But again, our focus in here will be on the disease-causing ones. So algae, these are eukaryotes. They have cellulose cell walls. They have different pigments to perform photosynthesis. Like you can see here, these obviously have chlorophyll, giving them the green color. These are usually found in some kind of aquatic environments. And being photosynthetic, of course, they produce oxygen and carbohydrates. They can be unicellular or multicellular. Unicellular forms are plankton, and multicellular forms are different types of what we tend to call seaweeds and things like that. They can reproduce sexually or asexually. We will talk more about algae in a future chapter, but you might be just kind of wondering, as far as algae goes and disease, you might be thinking, oh, I didn't think seaweed was dangerous or plankton was dangerous. And generally, they're not, right? In general, again, these are beneficial organisms, but there's a few, mainly of the plankton, that can produce some toxins that can make us sick. So when we do talk about algae in the future, we will mostly focus on those bad guys, on those ones that produce toxins. But we also get a lot of useful products out of algae, so we will touch on that in the future as well. So viruses. Viruses are interesting because they are not cells. So they're not prokaryotic or eukaryotic. They are acellular. So what are they? Well, they're basically nucleic acid, either DNA or RNA, that's enclosed within a protein surrounding called a protein coat or capsid. And that protein coat may additionally have a lipid envelope around it. Viruses, because they're not cells and they are generally not considered living, they don't meet all of the criteria of being a living organism, they cannot replicate outside of a living host cell, so they cannot replicate on their own. They're basically inactive if they're outside of a host cell. And then lastly, the multicellular animal parasites. So these, you can see these worms here, the heartworm, right? It's really disgusting. But you might be wondering, well, I thought microorganisms were microscopic, but I can see these. So this is an exception where we do cover these organisms that are not necessarily microscopic, but they have microscopic life cycle stages, like their egg and their larval stages, and that's generally when we get infected with them, is from their microscopic egg or larval stages. And we don't spend a huge amount of time covering these parasites, but we will cover them in chapter 12, and we will have a lab exercise where we do look at them. So again, these are eukaryotes, and they are multicellular animals. So they're not strictly microorganisms, but they do have microscopic life cycle stages. And again, you know, these are worms. You're familiar, I'm sure, with, you know, worms, earthworms, etc. Not all of them are parasitic. You know, minority are parasitic actually, but that's what we will focus on when we cover these, is the parasitic flatworms and roundworms, which are called helminths. All right, so that was the groups that we will look at, but how do these fit into the grand scheme of things when we talk about biological classification? This might be something you've learned about before. So life is classified into 3 domains. And the 3 domains of life are bacteria, archaea, and eukarya. And so then you can see how these groups that we just talked about fit. Obviously, bacteria is its own domain. Archaea is its own domain. And eukarya domain encompasses all those eukaryotic groups we talked about. Well, where's the protozoa and the algae? Well, they're here under the protist. Now, our textbook, we are using the newest edition, or you might be using an older edition, and that's okay. There has been some recent changes to the kingdom protista, but we're going to still kind of stick to the old school way and not get into the newer classification that they've introduced, because I think it's still kind of messy and being worked out. So traditionally, you have these kingdoms in the domain eukarya. The kingdom protista, which includes protozoa and algae within that kingdom, kingdom fungi, kingdom plants, kingdom animals. Okay, so I guess I must have, oh yeah, I did delete the first question because it was intended to be done in class as a poll, so I had to delete that one. So that's why this is numbered question number 2. You're actually going to be answering these questions within the recording on Canvas, so I'm not going to address the answers to the questions within the PowerPoints, but you will see these come up when you replay the recording, or while you're playing the recording right now. You will see this, okay? Okay, so moving on, so naming and classifying microorganisms. So again, something I hope you've learned about in the past, a little bit of classification that was established by Linnaeus, in the fact that all organisms have 2 names, a genus and a species, which is specific epithet, which means specific name. And when we use scientific names, there is a certain way that they're designated when they're typewritten. They should be italicized, or if they're handwritten, they should be underlined. The genus name, it always starts with a capital letter. The species is always completely lowercase. They are usually hard to pronounce, hard to spell, and that's because they're usually Latin-based words. And where do the names come from? Well, they may be descriptive of the organism. They may honor the scientist who discovered them. So again, the names can be quite varied, and you could see an example here. Escherichia coli was named to honor the discoverer, Theodore Escherich, as far as the genus name Escherichia goes. And then the species name was named for the habitat of the bacteria, which is the colon. Staphylococcus aureus is a descriptive name. Staphylo refers to clusters. Cocci refers to the spherical shape of the bacterial cells. And then for the species name, aureus refers to the gold-colored colonies. So when we use scientific names, the very first time that we use them, we have to write out the full name so that we're clear on what we're talking about. So here's an example with this sentence. Escherichia coli and Staphylococcus aureus are found in the human body. E. coli is found in the large intestine, and S. aureus is on the skin. So notice the first sentence we had to fully write out the names, but any subsequent use, you can use the abbreviation where you see that the first letter of the genus is used with the species name. So as I was pointing out, overall, microbes are not pathogenic. Most of them are not pathogenic. Really, it's the minority, but that's what we will be focusing on. But briefly, let's talk about, the benefits of those that are not pathogenic. So they provide a lot of benefits to our environment. Most of our microbes are out there being decomposers in the environment, helping to break down organic waste and recycle nutrients. Those that are photosynthetic obviously are very important as producers in the ecosystem. We can use a lot of bacteria to produce industrial chemicals, things like ethanol and acetone, and a variety of other chemicals that we'll talk about in the future. We also use the fermentative actions of microbes to produce various food products like vinegar, cheese, bread. And we also use microbes to produce a variety of products for manufacturing, medicine, and so on, like insulin, cellulase, vaccines, other medications, all kinds of stuff. Bacteria can actually also be used because of their decomposer, their degradative actions. They can be used to help clean up sewage. They degrade organic matter in sewage, and then they also, some of them can be used to detoxify pollutants such as oil, mercury, even cyanide and arsenic, things that are poisonous to us and most living organisms. There is some bacteria out there that can actually eat those toxic chemicals and break them down and neutralize them and make them, detoxify them basically. So the process of microorganisms breaking down something that is otherwise harmful or toxic is called bioremediation, because they're biological organisms and they're remediating or cleaning up or neutralizing that substance. So our textbook does have an entire chapter on microbial ecology. I think it depends on your edition of the book. I think the newest edition of the book, it's drawing a blank. I don't know where the book is, but I think it's chapter 28, if I'm correct. They do have an entire chapter. And why I can't remember the chapter is I don't cover that chapter. So if you're interested, though, you can definitely take a look at the chapter on your own. It is an entire chapter on microbial ecology. When I went to grad school at UC Riverside, I had to take an entire class on microbial ecology. It's very interesting. It's a really interesting area that would definitely focus more on that bioremediation and other interactions of microorganisms and the environment. This might also be covered in that microbial ecology chapter as well. Actually, not 100%, but I don't think it really comes up anywhere else. I guess maybe chapter 9, where we do talk a little bit about biotechnology. This might come up there. This is just, again, another example of some beneficial uses of microorganisms, specifically the fact that some microbes can be pathogenic to insects, and so we can use those microbes as an alternative to using chemical pesticides. It's a more biological or natural pesticide, and there's an example here with this Bacillus thuringiensis bacterium that can harm insects, but it does not harm us or the plants. One topic that you might be familiar with, just because it's in the news a lot, it's been talked about a lot recently, is the normal microbiota. So these are the normal bacteria, mostly bacteria. It can include other microbes, but majority of the time we're kind of referring to our normal bacteria that live on and in our bodies. There used to be the term normal flora. You might still hear that used, but it's not used as much. Mostly we use the term microbiota. So what is this normal microbiota? Or maybe another term you've heard to refer to this, the microbiome. So we have 30 trillion body cells in our body, right? That includes muscle cells, neurons, liver cells, brain cells. Well, I already said neurons, right? Whatever. All the cells in our body that make us up. And then you may have heard this. We have 10 times as many bacterial cells in our bodies. So we harbor 40 trillion bacterial cells in and on our bodies. Pretty crazy, right? So that's our microbiome. It's all of those bacteria that are living in and on our bodies. And so they live with us And generally, they're not harmful, and they're actually beneficial in many ways. And here's a few examples, but we will definitely talk about this more in future chapters. But we know from studies that they do help to maintain good health. They help us produce different growth factors and vitamins. They can help prevent us from getting infected with pathogenic microbes. They have huge roles on our immune system, but new studies even suggest they have roles in our mental health and just obesity. And I mean, the list goes on and on of the possible benefits of our microbiome. It's still being heavily studied, so we don't know everything about it yet. But things that we do know is that we begin to acquire our microbiome as soon as we're born. And basically, they live with us forever. So you really can't get rid of them. I mean, you can affect them. And I guess in some cases you can get rid of them by taking antibiotics, but generally it's not a good thing. You don't want to get rid of them. We can also pick up some transient ones, some that will colonize us temporarily, and then they may not last on us forever. And it all just really depends. It depends on our exposures from the time we're born. It depends on our hygiene and our habits. There's so much variation, and that's why it is just a huge area of study right now. So this is a really interesting video about the microbiome.
You do it every year, but it never gets old. Pack everyone in the car.
Microbes are everywhere. On your phone, in your water bottle, on your hands before you wash them, on your hands after you wash them, and literally everywhere else on top of you too. Microbes are omnipresent at any moment, and there is nothing we can do about it. So millions of years ago, we made a pact. We give them shelter and food, and in turn, they work for us. But the more we learn about this partnership, the more it looks like a Cold War. Inside our mother's womb, humans start out sterile. When we're born and traveling through the birth canal, billions of our mother's bacteria cover every single part of our bodies. This is an essential part of human health. Children born via C-section have a higher rate of asthma, immune diseases, and even leukemia. So our bodies do not only accept the invasion of microorganisms, they welcome it. Over millions of years, we co-evolved to make the best of our relationship. Mother's milk, for example, contains special sugars that are meant to feed and support certain groups of microbes, work as a decoy for others, and help to modulate the immune system. It takes up to 2 years until a healthy microbe community has formed. Every human has their own unique microbiome made up of bacteria, viruses, fungi, and other organisms. We have 3 categories of guests on and in our bodies. 1. Quiet passengers that do their own thing and are politely ignored. By being there, they take up space and keep more aggressive intruders in check. 2. Guests that harm us, but with whom we've learned to live. For example, bacteria that literally create acid that melts our teeth if we don't brush enough. They want to take up as much space as they can, and we don't want them to. But we can't get rid of them entirely. 3. Friendly fellows that our bodies want to have around. Most of them are a community of 380,000 billion bacteria from up to 5,000 different species that live in our gut. These gut microorganisms help us digest food and pull additional calories from things we can't digest ourselves. Unfortunately, our gut is also the perfect point of attack for intruders, so it's guarded by an aggressive army, our immune system. To survive here, our microbiome co-evolved with us to be able to communicate with our body. The most important part of that is to ask the immune system to not kill them. But they also have a real motivation to keep our gut healthy, so some of them produce messenger substances that help to educate the immune system, and others stimulate the gut cells to regenerate faster. But over the last few years, evidence has emerged that the influence of our gut microbiome goes much, much further. It might even talk directly to our brain. We've observed a few curious things. 90% of our body's serotonin, an important messenger substance for nerve cells, is produced in the gut. Some scientists think the microbiome does this to communicate with the vagus nerve, the information highway of our nervous system. Other examples are bacteria that stimulate immune cells in the gut, so they send a kind of alarm signal to the brain. Here it activates immune cells that help the brain recover from injuries. Since the brain decides when we eat, the microbiome is interested in a healthy brain. A new field of science is opening up here, and we're just on the verge of understanding how these complex systems inside our bodies interact. But we are starting to see how much our microbiome actually influences us and our behavior. Take depression, for example. Healthy rats fed microbes from the guts of depressed people began showing anxiety-like behavior and symptoms that look like depression. And in early 2017, a study linked the microbiome to intelligence by connecting a certain setup of bacteria in newborns with better motor and language skills. But it might also influence our daily lives. Tests with fruit flies showed that their microbiome influenced what kinds of food they craved. This could mean your microbes are able to tell your brain which food it should get them, although this is not a one-way street. The seed for our microbiome comes from our mother, but how it develops and changes is determined by what we eat. The organisms in our gut feed on different things. Some like fibers and leafy greens, others go for sugars and starches, and some love greasy fries and butter. Our gut is like a garden in which we constantly decide what will grow and blossom. If we eat healthily, we breed bacteria that like healthy food. If we eat a lot of fast food, then we breed fast food loving bacteria. Life is hard, so we can get trapped in a vicious circle. You have a stressful time and eat lots of burgers and fries and pizza. This is awesome for fast food bacteria. They multiply and take up space from vegetable-loving bacteria. But even worse, they send signals to the brain to continue what it's doing. This makes you want more fast food, which breeds more fast food bacteria, which makes you crave fast food, and so on. This kind of self-reinforcing cycle could play a huge role in obesity. But it's important to stress that you can fight this process and reverse it by eating healthily and breeding more good bacteria. Beyond weight gain, our microbiome has also been linked to other serious diseases like autism, schizophrenia, and cancer. One of the earliest symptoms of Parkinson's is actually gut problems. If your body is overrun with bacteria that harm you, there is often only one solution. You bring in an army of good guys. That's very easy. You just transplant some healthy poop. You do that by literally transferring poop from a healthy person into your gut. This method is already used to cure diarrhea that's caused when C. difficile bacteria take over a gut microbiome. But we just don't know enough about the complex interplay at work here yet. For example, a transplant from an overweight donor cured a woman's diarrhea, but contributed to her obesity down the line. This caused some waves, and another study tried to reverse the effect. Poop transplants from slim people to obese ones gave them a more diverse microbiome and made them less sensitive to insulin, both things that also happen when people lose weight. We need to do a lot more science to really understand how our microbes make us healthy or sick. But whether we like it or not, we need our microbiome and it needs us. We'll never have our bodies to ourselves, but we have gained a powerful ally, if we can just keep the peace.
See if we can get out of here and go back. There we go. So very interesting video. So this is just talking about the Human Microbiome Project, but we kind of need to move on. So we will just move on to the next topic, which is the history of microbiology. And I'm going to go over this pretty quickly and pretty briefly. You can definitely read more about these scientists and their discoveries in the textbook. We actually do have another YouTube video to watch. This one will be shorter than the last one.
It's your favorite family tradition to drive to that familiar beach house with familiar faces.
When animals first evolved, they did so in a microbial world. They did so on a planet that had been home to all these organisms for billions of years.
This is Ed Yong. He's written a book about the microorganisms that live inside us and all around us, and that predate us by a lot.
If we condense all of Earth's history into a single calendar year, then life emerged around about March. And multicellular life, all the organisms that we're familiar with, only really came up in October or so. And humans emerged very, very recently indeed. So we're sort of just the icing on life's cake.
From the time humans evolved, bacteria and other microbes would have colonized every nook and cranny of our bodies, just like they had other animals. And they lived with us like that for some 200,000 years, until eventually someone looked close enough to notice. That person was Antony van Leeuwenhoek.
So this is the late 17th century in the Netherlands, and he's not a trained scientist, he's not a scholar, but he has two things that are really important. One is an insatiable curiosity, and the other are really, really good lenses, which he grinds himself.
At that point, microscopes had been around for decades. In England, Robert Hooke had published a book of observations that was a popular success. It introduced the term cell for the pores that he saw in a slice of cork, because they reminded him of the cells in a monastery. But bacterial cells are much smaller than plant cells. While Hooke was using a compound microscope that could magnify 20 to 30 times, Leeuwenhoek used single-lens microscopes that could magnify up to 260 times, though you might not expect that from looking at them.
It's really like two brass rectangles that sandwich this tiny, tiny glass lens, the spherical lens between them, but it basically looks like a door hinge.
The whole thing was just a few inches long, and the lens was as small as a grain of rice.
And he would have held it really close to his eye. He would have had to basically press his face up against this thing and look through the lens. It would have been deeply uncomfortable and very straining.
To understand why his lens was so small, you have to know how magnifying glasses work. Like any other lens, they make use of the fact that light bends when it enters glass at an angle. That's because light travels more slowly through glass than air, and so the portion of the wavefront that enters the glass first will slow down, while the other part continues at the faster speed. That's what makes the light change direction, both when it enters and exits the lens. Now, if you put an object close enough to the lens, the bending light creates a virtual image that's not the size of the real object, but the size it would be if the light from the lens had traveled in a straight line all along. And if the lens is more curved, those lines get even steeper, meaning greater magnification. So Leeuwenhoek's lenses weren't just biconvex, they were spherical. And he knew that he could make that curve even more drastic by shrinking the whole lens down as small as possible, even if that meant he had to get his eye uncomfortably close to it. And with that tiny bead of glass, Leeuwenhoek opened a peephole into a whole new world. He looked at his blood and discovered blood cells. He looked at his semen and discovered sperm. He looked at different samples of water and saw algae cells, protozoa, and bacteria. He called them little animals. He went on to peer into his own dental plaque, and he found bacteria there too. And he described what he saw in letters to the Royal Society.
He's the first person to see bacteria. He is the only creature, let alone human, he's the only organism in the entire history of life on Earth to actually see the things that have been the dominant players in life's history. And I think the great thing is that he wasn't disgusted by it. He saw tiny living things living in his own mouth, and he thought they were cool.
It would be nearly 2 more centuries before microbes were linked to disease, and even longer before we understood that our microbes help us digest food, that they train our immune system, and help crowd out harmful bacteria. Now microscopes are much more powerful than Leeuwenhoek's were, but these days it's a different tool altogether that's enabling researchers to explore how microbes affect human health.
Just like Leeuwenhoek found microbes because he had the right tools, now we're seeing that the microbiome is important because we have the right tools. Because we have the ability to sequence the DNA of microbes, to identify them by just scooping up a sample from the environment and looking at their genes.
If you were to take all the water out of your poop, 25 to 50% of what's left is bacteria, both dead and alive. So if you were feeling sad that you can't see the microbes that make a home on your body, well, it turns out you kind of can.
They're known as Marty's
Okay, so that was a really cool video, and definitely talked about Van Leeuwenhoek, so I'm not going to cover him, but you definitely need to know him, so you can read further in the textbook and get more details of these scientists. But I want to move on to spontaneous generation because this is an important topic that you will need to know and distinguish spontaneous generation and biogenesis. So spontaneous generation was a hypothesis that was long held by mankind. And basically it was the belief that life just spontaneously just generated. As long as there was a vital force, which most people thought was air. As long as there was air, life could just poof. Frogs could just come out of ponds. Snakes could just come out of holes in the ground. Flies would just emerge from manure and corpses and things, right? So they hadn't observed reproduction of, especially the smaller the organism, they hadn't observed it. But even things like frogs and snakes and stuff, they generally believed they just spontaneously came about. But then as scientists started making a lot of discoveries, especially with the invention of microscopes and the ability to see things that hadn't been seen before, scientists started thinking that spontaneous generation just couldn't be. That can't be how it is. It has to be, more scientific. There has to be an explanation for where life comes from. And so biogenesis was that explanation, that living organisms arise from pre-existing life. And this also tied into a theory called the cell theory, which said that all living organisms are made of cells. And then that ties into the biogenesis, that all cells must come from pre-existing cells. And so let's watch this brief video on spontaneous generation.
Distance has been on all of our minds lately. That we've been too close to
stuff that wasn't alive. Worms just seem to appear in the wood of ships. Mushrooms seem to grow out of dead logs. Mold grew on old bread. This idea is called spontaneous generation, sudden life. It's an interesting theory. But it turns out to be wrong. Dead wrong. See, a French scientist named Louis Pasteur showed that all life comes from other life. He boiled broth, soup, in a bottle to kill anything that might be living inside. He fitted the bottle with a long neck so that nothing from the air could fall into the soup. And sure enough, nothing grew inside. But when he took the neck off, things started to grow. Mold and bacteria, all kinds of stuff. See, there are things in the air that are at a stage of their life cycle that's invisible to us. When they fall into soup or on a piece of fruit or on a piece of old bread, they can grow big enough for us to see. All life from life. Omne vivum e vivo, we used to say.
What?
Yeah, this is true for you and me and mold on your breath. I know what you're thinking. It's going to be really funny if I took a bite of this moldy bread. I'm not going to do it, because there's living things there.
See for yourself whether spontaneous generation happens.
Did you say spontaneous generation?
That's right. I said spontaneous generation. You take 2 plastic jars like these, and you run them under really, really hot water.
Don't burn yourself.
Then you put a couple pieces of banana or other fruit in both jars. Quickly screw the lid on one jar really fast, but leave the other jar open.
Gotcha.
Let the jar sit undisturbed for 2 weeks. Better put your jars outside at this point. If you're lucky, you'll see tiny maggots in the open jar.
You were lucky.
That's the first stage in their life cycle. They're baby flies. In the closed jar, no flies can lay their eggs on the fruit. Zip, none. No life cycles happening in here. For a life cycle to get rolling, you need a little life to start with. Life can't just appear out of nowhere.
Let me write that one down.
You do it every year.
Okay, so that video was very poor quality, but it's the only version of that Bill Nye episode that I could find. It was a very poor quality video, so sorry about the quality, but I think it really got to the point of spontaneous generation and biogenesis. So you will need to know more about Pasteur's experiment, as Bill Nye was showing, that showed, basically disproved the theory of spontaneous generation and supported biogenesis. So as Bill Nye was explaining, he took some broth, he put it into a flask. It had this long bent neck, which they call an S shape or swan neck flask, which was so steep that microbes could not transcend up the neck of this flask. So he boiled the broth within to sterilize it, and it remained sterile. Even though it let air in, it didn't let microbes in. So with this work that Pasteur did, he basically showed that there's microbes in the air. They can get into substances and contaminate them and grow if they're able to. But if they can't get in because they're blocked, then the substance can remain sterile. We might wonder, well, why not just put a lid on it? Well, the problem with that was that whole vital force. A lot of scientists would get upset with the other scientists. There was this huge debate of spontaneous generation and biogenesis. And they would get upset if people sealed the broth or whatnot in their experiment, because they would say, no, you can't seal it. You need air. You need the vital force. Which is not entirely true, as we'll see in the future. We'll talk about anaerobic microbes that grow in the absence of air. But to satisfy those who thought that air absolutely was required, that's why he designed this flask that did let air in, but did not let microbes in. So I kind of just told you the gist of that, why he bent the flask that way to let air in but keep microbes out. The flask showed no signs of life. Even still, it's in the museum, in the Louvre Museum in Paris, France, still sterile. So you might also know the name Pasteur from pasteurization. So Pasteur, actually, he's the father, or the godfather, if you will, of microbiology. He made a lot of discoveries in microbiology, so we will talk about him a bit. But definitely pasteurization is something that you're probably familiar with. So he was French. He lived in France. Of course, wine is very important for the economy in France. And so the vintners, the winemakers, came to Pasteur and wanted to know why sometimes their wine would turn into vinegar. And so he investigated it. And so we know that the way that wine is made is through a process called fermentation. where the sugar in the grapes gets turned into alcohol. And fermentation is done by microbial action. The microbes are fermenting the sugars and they make alcohol as an end product. Well, there's other microbes though, specifically some bacteria, that can then take the alcohol and further ferment it or spoil it and turn it into acetic acid or vinegar. So in some cases, wine would end up getting turned into vinegar, and of course the winemakers didn't want that. They wanted wine, not vinegar. So they asked Pasteur to figure out why and how that was happening and how to stop it from happening. So what he figured out was if he heated the wine enough to kill the bacteria, but not too much, you can't just go boil the wine or the alcohol would evaporate off and now it's not wine anymore. It doesn't have alcohol. And so he said, okay, just enough heat, a small amount of heat, kill the bacteria, but not destroy the alcohol content. And sure enough, it worked. And it preserved the wine. It kept the wine from turning into vinegar. And the process was named pasteurization. And we use it not only for wine, we use it for milk, we use it for all kinds of food and beverages. So pasteurization is the application of high heat for a short time to kill either spoilage or harmful bacteria. So during this time of Pasteur's work and many other scientists that you'll see in the chapter, they called this the golden age of microbiology. There was just tons of discoveries being made in the fields of microbiology during this time. Another one that Pasteur definitely contributed to is called the germ theory of disease. This is where they finally started realizing that disease was not caused by being punished from God, because that was kind of the commonly held belief at the time, that if someone got a disease, it was because they had done something wrong and God was punishing them. But they started realizing diseases were caused by microorganisms. And the first ones they actually discovered were in silkworm, because silk was a very important commodity at the time as well. But also they started making discoveries with human and other animal diseases as well. Semmelweis, we will talk about him and hand washing in the future. But he basically was the first to advocate hand washing to prevent disease transmission of new mothers. So puerperal fever, also called puerperal sepsis or childbirth fever, was a very common disease that killed women within a few days after giving birth. And it turned out through Semmelweis's investigation that women were getting this infection from the doctors who delivered their babies, because the doctors usually were doctors in training, interns at the time, and they would spend all day working on their anatomy, working on cadavers, and believe it or not wearing gloves and not washing their hands except maybe wiping off the gunk on a rag, and then go deliver babies. And so they were transmitting bacteria to the woman's uterus, basically, during the delivery of the baby. And so within a few days after childbirth, the woman could get a horrible infection in the womb, in her uterus, and basically die. So he advocated hand washing, and lo and behold, the survival rate for women increased quite dramatically. The rates of that childbirth fever dropped. Joseph Lister is another scientist you may have heard of. Well, maybe not of him, but the product that was named after him, right? Listerine. So he was the first to use a chemical disinfectant during surgery. Koch and Koch's postulates is someone that we will talk about in a future chapter, but again, he was another one that played an important role in the germ theory of disease, and he developed these postulates, these steps that could basically show that a disease was caused by a microbe. Jenner is another one that we will definitely see in the future, especially when we cover vaccines. But what he observed was that milkmaids, women who milked cows, did not get smallpox, which was a very bad disease at this time. They would get cowpox, though, on their hands from milking cows. Yet they seemed to not get smallpox, and therefore milkmaids were known to have very beautiful skin, because people who got smallpox and survived would usually be left with scarred up skin. So he kind of saw this correlation and decided to test it. And so he took some fluid from a smallpox lesion, and then he took it from a lesion on the milkmaid's hand, and he introduced it to a little boy. And then a few weeks later, he challenged that little boy with smallpox. And thankfully, the little boy did not get smallpox because he got protected from being exposed to the cowpox. And thus began really the first vaccination campaign. And it was so effective that we actually eradicated smallpox. So again, here's a question that you're going to see right now within the video. So the first synthetic drugs, so during this time, especially once they discovered that microbes were causing disease, of course they wanted to know then, well, can we cure it? How do we kill the microbe? How do we get rid of the disease? So they started investigating a lot of chemicals, but the bad thing with chemicals, as we know, is they can be toxic. So they were looking at things like arsenic. There was a drug called Salvarsan that was used to treat syphilis, but it did have a synthetic arsenic as the component of the drug. We also, of course, at the time looked at natural products that worked, like quinine from tree bark to treat malaria. And then we synthesized what we kind of call antibiotics, although sulfa is not necessarily a true antibiotic, but we did synthesize sulfa drugs in the 1930s. And then soon after, kind of during that time, but it took a little longer to get it through clinical trials and get mass produced, penicillin was discovered and introduced in the 1920s through 30s and finally released in the 1940s. So interesting thing about penicillin, it was discovered by Alexander Fleming by accident. He was working with bacteria and his petri plates got contaminated with mold. And what was a lucky accidental discovery was that mold called penicillium that you could see here on the plate, that wherever the mold grew, the bacteria didn't seem to be able to grow. And he realized the importance of that maybe the mold is producing a chemical to inhibit the bacteria. So he studied it, he isolated the chemical, named it penicillin, they tested it, it seemed to work, so mass produced it, had to go through clinical trials, obviously, and the rest is kind of history. These are just terms for the different studies within microbiology: bacteriology, mycology, virology, parasitology, immunology. Pretty straightforward. Interesting story on this medical symbol, right? You may or may not have heard before about it being due to the fact that there's this worm infection in which the worm has to be removed from the tissues of the infected patient, but it has to be removed slowly over time. So every day a person would take a stick and kind of wind the worm around the stick and slowly remove the worm without breaking it off. Because if the worm gets broken, it causes a horrible inflammatory reaction in the body. Another topic that we'll definitely touch on in future chapters as well is biofilms. So biofilms is when bacteria grow in a slimy film, and it's usually mixed. It doesn't have to just be one bacteria. It doesn't even just have to be bacteria. You can have algae and fungi and protozoa and bacteria. Can be a variety of microbes growing in this biofilm. But again, our focus is on bacteria, so mostly we will refer to biofilms as those produced by bacteria. Everyone has encountered a biofilm before, maybe just didn't realize it. If you ever went into a pond or a lake or a stream or something and you've stepped on slimy rocks, that's a biofilm. If you've ever had to take apart your pipes in your house because they were clogged or something, and you noticed all that slimy stuff on the pipes, that's a biofilm. If you have plaque on your teeth, as we all do, that's a biofilm. But the unfortunate area where biofilms can grow, of course, plaque and all that's not good either, but kind of a newer revelation is that biofilms can grow on medical implants. People who have long-term indwelling catheters and other medical implanted devices, unfortunately, we have seen that biofilms can grow on those and can even lead to fatal infections in some cases. The bad things about biofilms is that they are resistant to antibiotics. They're harder to disinfect. So that is an issue. And again, we will talk about them more in the future. That's just a brief introduction to them. Here's another question that we will see come up while you watch the recording here. And lastly, another question. Or I shouldn't say lastly, because I think there's one more after that. Okay, so these will all be in the recording on Canvas.